think I'm like,
Look at
wow,
that.
that tail is fabulous.
So if we find a grenadier, we'll zoom in on it for you, Chris, and then you can see really see the difference on the tail.
Yeah, that would be awesome.
Hear that, grenadiers? Come on out right now.
So we may, depending on what dives next, we might be heading to south of Santa Cruz again for either the next dive after this one or the one following that, depending on weather, as I understand it. So, Tom, you might get your wish. Be back in the same habitat.
Yeah, we looked at Anacapa and we just didn't happen to see any, because I've seen them, now that we know what they are and where to look, I've seen them like through the express cruise, gosh, all the way from, did we see it on Sphere Drop? Anyway, from down in Southern California up to about Mendocino.
Okay.
So, and they would really previously only been known from a few collected by trawl. And so this was good to see them, and they always were in the same type of habitat, similar depths, like 400 to 800 meters.
Also, science, yeah. So I'd say this because we're like 150 meters or actually 200 meters from the last transect, but that slopey area is just about 230 meters away from us. So if you're going to want to transect between now and the end of this watch, I might recommend that we do it before we hit that slopey area so we could explore it. But
that sounds like a great plan. Let's set up for that, and we'll run it until we can't.
Roger. Okay.
or it ends.
Yeah.
One or the other.
Roger that.
Yeah, I've been talking about the ruby nose, so hopefully we'll get one in here so that everyone will be able to see it.
Let's come down a little bit, and then we can start it.
Roger.
Has that made it into your PowerPoint fish ID guide? I'm about to call up.
All right, Ben.
Okay, great.
It should be in there.
This is T7.
Oh, thank you. Thank you, Mary.
Yeah, I'm glad. That's what I have too, so yay.
All right, stepping forward.
There's the Chimera.
A grenadier.
Grenadier.
Grenadier?
Yeah. Whoops.
But you can see, Chris, as it comes down to a point and not a normal fish-looking tail.
Yeah, that's very convenient. I like having one after the other so I can get my IDs right. I appreciate the effort on the part of the Nautilus team.
And the grenadier for showing up.
And thanks also to the Grenadier.
Sounds like an Academy Award there.
Yeah.
And we have a sea pen coming up too. Look at that. Fish and a coral, all within the first minute or so of the transect. That's awesome. Guess we could include the crab too. So.
I did not find
There's a
your
There's a baby fish.
friendly. Where's that? Oh yeah. The ruby nose. I did not find that in the PowerPoint.
Well, let me see what I have in this one.
Time for an update. Next edition.
Well, it may be updated and you just may not have them.
Oh, that's true. Yeah, mine says 9/29/10.
Mine says 10:08:20.
I'm going to come down on the delta a bit there, please.
This is the one I downloaded from our guide folder on the.
Really? Did I not put the newest one in there? Oh, I might have to do that. Well, I'm seeing it looks like we have a little coral down there next to the anemone. So you can talk for a minute, Chris, while I add this to
Take care of that.
section.
I will take over. So this is, I'm assuming T7 for the 7th transect.
Correct.
So as we're exploring Santa Lucia Bank, we're doing a combination of timed or distant transects over approximately 200-meter distance, where we're quantifying all the fish and corals and sponges. We'd be able to use that for comparisons with other areas we've surveyed up and down the West Coast to assess areas where there's new species or species range shifts, to assess areas that are higher or lower in diversity of different taxa, and better understand the ecosystem, the biodiversity, and the fisheries that are sustained by it. In addition, in between transects, we're in what we call exploration mode, where we are just looking for any new species, new taxa, species that might look differently. We'll collect them and study them when we're back on land, and just try and characterize this area. This area, if you're just tuning in, is Santa Lucia Bank off of Morro Bay, or the Santa Lucia Escarpment just off of Morro Bay. We'll be here until roughly, I think, 3 p.m. This afternoon, maybe earlier, depending on what time we have to leave the seafloor to get the ROV back on deck. That's 3 p.m. West Coast time, California time.
Yeah, I think we're estimating that it will have a 3-hour ascent.
Yeah. So we're going to be on deck at 1600. So we're going to probably come off bottom at 1 p.m. Yeah.
Okay.
But blue water's cool too.
You can still.
It is. We're going to see your giant squid then.
Yeah.
All right, Chris, I put the new fish guide updated in there, and it'll have your ruby nose.
Excellent.
And for all of you in the van, if you want to see that, we have the folder. There's the ID guides or deep sea taxonomic guides in there.
Awesome.
I have my own PowerPoint that I've made up of all the fish that we've seen.
Very cool. Yeah,
Super
thank you.
cool.
That crab is covered in mud. That's got to be a boy crab. He doesn't care how he looks. I had 3 sons, so I know.
Kim, you want to come up on the
I guess
deck?
I should say I have 3 sons. I still have them.
Ben, I think video's trying to call you.
Video?
I like that you put the older fish guide up there originally just to keep us one step behind you, Tom.
Actually, I think Jan uploaded it, and I didn't pay attention to the date because it had her name on it, and so I just thought that she put the newest one, but and there's a sea spider, a pycnogonid.
Big one.
Yep. There's a lot of these deep sea critters and small ones that if you blow them up, they would be just such great horror movie critters.
Oh, this has a lot more. The ruby nose. All right. I've got my homework cut out for me.
Now that we saw that first cucumber, we're seeing more.
At what depth do you stop seeing the Sebastes?
So most of them will stop around 500 meters. And then there's a couple species that'll last to 650. But generally about 500 is where you start, like if you're working up a slope, where you start to assume that you'll start to see them.
Okay.
It's like, I don't even know what depth you'd get to thorny heads, what their deepest is, because you see them at 1,000, but now we're at 3,000 or so, and we don't see them, so.
Yeah, definitely significantly less.
It's still on the
Less
same
diverse
depth
down
contour.
here.
It'll be going down slope if any, right?
Yeah, we're good.
Oh, if we do a eastward move, then yeah.
Yeah. Exactly. I'm just thinking of stopping the ship moves a little bit before so we can get that momentum.
Yeah, we can stop it and see where we settle, and then we can swing over.
I think after this move.
Sure. How much longer we have on our transect?
Yeah, we have 90 meters left on the transects.
Roger.
Woohoo, halfway, yay.
Woo.
I find I always ask when we're at 90 meters left.
But, yeah. It's when the restlessness sets in. Yeah, no.
Or that's just what he says every time.
Yeah, exactly. We've actually just been driving in circles.
Did you break out the chocolate-covered espresso beans?
Ooh, how'd you know?
I'm just figuring it out.
We usually bust those out around 6 a.m.
Did we seem more lively?
Yeah. Our personalities have dramatically changed.
Time to get up. I hear the alarm. I'm seeing a little bit more rock kind of covered in the mud, so it may be changing.
Hope so. It would appear that way on the map a little bit.
Well, the ship already knows because it's there. So.
Right, exactly.
Let's call ahead.
Yeah, I work with a two-body system sometimes where we tow kind of a weight right off the seafloor, but we only have depth, good depth from the ship. So depending on the layback, it feels like, you know, extrapolating from something way ahead. What does the ship say?
Sorry, giving you a tug there, Kim.
Hi, folks. This is Pete Curtis. Do you copy?
Yep, we copy.
Yep.
Pete, what's up?
Is that Marley?
Yeah, hi.
Hi, I'd recognize that enthusiastic voice like anywhere.
Anywhere. Yay, I'm so glad you're here.
Thanks for having us. I just wanted to check in at the beginning here. I'm going to be hanging out with my students who are tuning in as we speak. And we're just kind of watching you all on nautiluslive.org. We had discussed sort of having about 30 minutes or so at the end to kick around some questions. I'm not getting the sense that you're all overwhelmingly busy. Would it make more sense, or are you open to us sort of chiming in every now and then?
Yeah, this is Tom Leidig, and for us right now, we're on transect. We have, I'm guessing, about 50 meters to go. And since we haven't been seeing much, we're just going to be exploring after that, for probably till the end of the dive, or the end of our watch, which will be 8 o'clock. So that'd be a great time to throw out questions, comments, whatever you guys want.
Outstanding. Thank you all very much. It's a delight to hear your voices, and I hope you all have a smooth ride out there.
My ride's really smooth. I'm sitting in my living room.
But you're in California, right? So you never know.
That is true.
That's right. Yeah, if things start shaking here, I don't know if I'm going to think I'm on a boat or hit the decks. But
I was an undergraduate at UCLA during the Northridge earthquake, and I had a pretty big fish tank at the time, and during the earthquake, that thing started sloshing, and the only thing that came to mind was to save my fish. until I looked at the tank and thought, this thing's going to kill me. So yeah, I miss California. It keeps you on your toes.
Yeah, we haven't had a big one in a while. I went through the Loma Prieta one up here in Santa Cruz. What was that? 89, just about 31 years ago. And that one kept us all on our toes for a while. Here comes a giant cusk eel coming through right now.
30 meters.
Roger that.
Yeah, thanks again for letting us join you. We're going to sit back and watch you tell us about what you're coming across. And again, I'll gently chime in and see if you all have a moment to answer any questions as they come up. So thank you again.
All right, sounds good.
Sounds great. Great to have you. Well, they're tuning in at just the right time, I think. For the last previous hour or two, we're primarily over just soft bottom and sediment. More complex habitats that have some of those rocky ridges that are reefs tend to have a little bit more in terms of organisms associated with them, a little bit higher biodiversity just because of the mix of habitats. Some animals prefer to attach, like deep sea corals or sponges, more likely to be found attached to hard structure. So when we start seeing stuff like that, now we've got our eyes open for a whole other set of species. In terms of the fishes, and I've been learning a bunch from my colleague on the phone right now, who's an expert. We've been seeing grenadiers, cusk eels. He just mentioned that's the last one we just saw was a cusk eel. And fish of the family Moridae. And I just learned to tell the difference, one of the differences between grenadiers and cusk eels. The grenadiers, if you look at the tail, it sort of comes to that really sharp point versus having a more typical fish-like tail that you would see in the cusk eels.
Glad you're learning. By the end of this, you'll be the expert, and I won't have to say anything.
I hope I get my certificate in the mail.
Okay, as soon as I print it up.
We can see as the lasers are just kind of going over that soft bottom. If you look really closely, it's not that there's nothing there. There in fact are slightly buried brittle stars.
And right under the lasers, there's a sea spider.
Sea spider. Little sea spider.
Just went by.
And that pink area that's just below the lasers, that's the anemone. So every little blip is often something. It could be small, but we're paying attention to all of it the best we can.
The caveat, the best we can.
The transects we're doing are about 200 meters in length. And that video footage we store and will be reviewed by an expert who will annotate that video and let us know how many of different species we saw, corals and sponges and fish. There's a little fish on the bottom right.
Yeah, so this is 200 meters now, so we can call that. Yep.
Roger that.
Hi, folks. This is Pete. What are we passing there on the screen? Is that just a rock or little outcropping?
Yeah, we're just now starting to get into a rockier area.
We're still doing a little bit of a drift. Sorry.
No problem. Yeah, if we could go up to the coral up above us right now and just zoom in on it. So we just came out of the mud area. We're getting into an area of a little bit more rocks and hard habitat.
Let's do 30 meters.
Roger that, thank you.
140. Oops.
And this is a heteropathes black coral right here. And to the left, there are stalked crinoids.
Did you guys still want to zoom on this guy?
Yes, please.
All right, go ahead, Marley.
And it's called the black coral because its skeleton is black. So a lot of times in science, the obvious common names come from obvious features of the creature. That's good, thank you.
Sure thing, combine.
Oh,
Go ahead and combine, Marley.
You can see the little red brittle star just below it to the left.
And now that we've finished the transect, we're doing some exploration. So we're going to go around this area and take a look at some of the things. Oh, that's an umbellapathes. I'm sorry, I called that a stalked crinoid. That's also a black coral that was in the left. Now that I get a better look at it. And we do have a yellow crinoid coming up almost under the lasers now. And as we get to this harder habitat, you'll see more animals that live attached to the rock.
Yeah, Jess, after this move, I'm going to just go due east.
Roger.
It's going to be slightly upslope, but
Roger
good.
that.
So is the ship stopped now? Are we just hanging, or are we actually moving?
Yeah, we settled out a little, and then I'm bringing us just a little bit more south. But I'm going to call in for an move to the east. So we're targeting, yeah, this.
We're just staying in the orange area.
Yeah.
Okay, roger that.
Yep.
So for you, those of you at home, we have another screen that has the navigation on, and we were looking at that. So you probably don't see an orange area on the screen you're looking at now, so that you could be confused with that. There's a green sea urchin up near the top of the screen. Which we've been seeing off and on. It's interesting that we've only seen these guys really around the hard substrate today, not on the mud when we were cruising along.
There's a shrimp over to the left of it. And a sea spider just below it. So those lasers, if we didn't mention before, those are 10 centimeters apart. So that gives you a bit of a feel for how big that sea urchin is. Just a little bit bigger than that, maybe 12 centimeters or so. by having the lasers on, that's the only way we can tell what scale or size something is, because we can't tell necessarily exactly how far we are from it. So immediately that gives us a quick check to see if something's particularly large or small. Tom mentioned the other screen that we were looking at. So when we choose a place to dive, one of the things that we use to select it is depth or bathymetry. Many of these places in the ocean still haven't, oh, look at that, Sunstar. Many of these places in the ocean are still unmapped. We don't even know what depth they are. They'll have been mapped at a very coarse resolution using lead line data when they would drop over weighted line from a boat and figure out how much line they had to drop over before it hit the seafloor.
Right here before we go.
Yeah, sure. Which one?
That's okay. It already went under. It just slightly back, and then straight below is the tall one.
Sure thing.
As long as we didn't dust it. Right there.
Hi, Marley, go ahead.
So this one only has one row of polyps, whereas the last one we looked at had 2 rows. So I believe this is a different species.
Should we collect?
You know what? Why don't we just collect this one?
Come wait, Marley, please.
Just wants to do something other than fly.
Oh, I'll take anything.
To mix it up.
Well, since it's pretty skinny and thin, we can take a hair sample from the top and then suck it up.
Yeah.
Yep. Okay, Raj.
Sounds good.
Sure.
Can we get a close zoom in at the base before we do anything, just to check it out?
Oh yeah, sure thing. I'm just gonna sit down, and then we can do all of our biddings. Ben, do you want to back the ship up a little bit?
Yeah, I'm trying to see. I can do that.
All right. I was a bit into the vehicle now. Yeah, he's into the vehicle a bit there. Oh. Hi, Marley, you want to zoom in on the base, please? Maybe we can get the porch light on too, and that might illuminate it a bit better. Ben, you want to keep an eye on the muzzle, please?
All right, that's full zoom.
Raj.
That's good, thank you.
All right, go ahead and come wide, please. Okay. Raj.
The claw.
Did you say you were going to snip and slurp this?
Snip and slurp, Raj. Let's get a little smaller. Mm-hmm.
Another spaghetti twist.
Now we got it.
Yay.
All right, Kim, go ahead and give her a flush. And have there been any suction chambers that have been used so far?
Yes, 1, 2, and 3 are all used.
Raj.
So are we going for 4?
Yeah, we can go for 4. All right, Kim, I think that's pretty good. Stand by. Hey, Marley, you want to zoom in here a bit, please? That's great. Raj. All right, Kim, go ahead and try sucking it in there. Yeah, she'll go now. Nice. All right, come on, Marley. Yeah, nice. Nice, thanks, Kim.
Good job as always.
That was a fun one.
That's what we're here to do, is just do fun things. Even when we're on the mud.
Yeah.
It's still fun.
100, Raj.
And just to
Go ahead
check,
and
that
dial
was
it in.
sample 70, correct?
Yes.
Thank you.
All right.
So for people tuning in, we've just collected part of a deep sea coral. If you've been paying attention, we've had to be very careful about noting we only have so many storage compartments, and so we don't want to get our samples mixed up. We want to know what was collected where, so we can match the sample with the video and still footage that we take of each sample before collecting it. And we have different ways of collecting. We have a storage box where we can put heavier items, and lighter items we can suck up, which is what you saw just there after we clipped the piece of coral.
All right.
So now we're continuing on the ridge.
Yep, looking for more diversity.
Folks, this is Pete. We have a bunch of students out here who are enjoying the show, and I'd have you know that Grace Matthews is a student at Harvard who's working in Rob Wood's lab, and that's the lab that produced the soft manipulator on the front of the vehicle.
Cool.
Oh, that is really cool. That's been important. That's been essential to our ability to collect these tiny little soft critters without crushing them with that giant claw you just saw.
Could we zoom in on the octopus? Oh, no, never mind. That's an anemone. Caught me for a second, thought we had an octopus. Looks like there could be suckers around the edge, you know?
Yeah, good.
Octonemone.
Can we actually zoom in? There's a sponge to the left of the yellow coral. It's a little tiny barrel sponge.
Oh, yeah.
So, and can we get the lasers on?
Sure. Would you mind put on the lasers, please? All right, Marley, go ahead.
We haven't seen many sponges this dive, so this is one of the few. And you can also see the brittle star well, and there's a little coral on the underhang.
Go ahead and come
That's
a little
good, thank
wider,
you.
Marley. Sure. Go ahead and come forward.
So we're just going along this rocky area looking for different corals and sponges and some other inverts to sample, and just exploring right now. It's a new area. No one's ever dove this escarpment before. So this is how we find out what lives here. Can we zoom in on the fish for everyone?
Sure thing.
Chris, you get to tell us what it is. No pressure.
It's not a grenadier. It's
All right, Marley, go ahead.
Skeleton head. Such a nice shot. So that's a giant cusk eel. And can we have the lasers back on?
Roger. Go ahead and come wide, Marley.
That'll give you an idea of the size of it.
Actually put the lasers on it if you want.
Sure, we can put the lasers on.
That's like
That was
40
half
to
the
50
lasers.
centimeters or so.
Yep, there you go.
Big one.
Gotta wait till he straightens that tail out.
By the way, I did have the idea correct. I was just on mute. Yeah.
I guess we could give you that, but
partial credit.
Sounds a little fishy.
Nice.
Oh.
Nice.
From the back row, married to ladies and gentlemen.
Not too early.
Yes, Mary.
I gotta think of jokes ball back here.
The trick to all this, you find out that you've been talking to yourself for 5 minutes.
been there, done that. And that's usually when you have the best explanation of something, too.
So,
don't feel bad. I one time had an entire text conversation with myself on my phone.
So I mentioned the mapping work earlier, the lead line data, before we sampled that coral. But now we have sonar data, much more high resolution, what we call multi-beam sonar, where we can go over the area and collect much more high resolved information to tell us what's going on with the seafloor, not just how deep it is, but is there a slope or a ridge? Is it complex or relatively flat? And also, you get two pieces of information. You get the speed of the return, and has to do with depth, and the intensity of the return, which has to do with is the sound. Sonar is a sound pulse. How strong is the signal coming back, which tells us something about if we're sampling on hard bottom or soft bottom. And so that can also be key to guiding where we're going to put the ROV down. The ROV pilots want to know, are they going to be going through very simple habitat, or is it much more complex, and at what level? They have to be very careful so they don't damage the equipment. What does that environment look like optimally before we go down?
Can we zoom in on this white? I think it's a fish tail.
Sure. Oh,
There it is. Ooh,
oh, come back, come back. Come
different
back.
eel.
That's a fast swimmer right there. Let's see if we can find it.
I see a shadow go away.
Go, Jess, go.
No, I think the fish is.
Too high.
See it in Argus.
Yeah, you can see it in Argus, but he's just swimming vertically now. Probably not worth it.
Smart move, eel.
Pictures today.
Well, we got to see his tail. That's all we needed.
You sure that's all you need?
not pointy.
We weren't on transect, so that's all we need. If we were on transect, we'd have to chase him down, find out who he was.
I think that'd be considered a vertical transect.
No, that's a little later today. We'll do one of those, 200 meters up. And then you
For
get
those
snow
tuning in,
and
we're at 2,900 meters depth, surveying at the Santa Lucia Escarpment off of Morro Bay in California.
Hey folks, this is Pete. We've got about 24 of our deep sea biology students on now. I have them on mute. I just wanted to ask you all if you were open to kind of maybe in a minute or so going around the room and just kind of sharing a bit about yourself and what your specialty is and, I don't know, some fun fact. Does that sound okay to you all?
Works for me.
Great.
Since we're just exploring, yeah, it's a great time.
Perfectly acceptable answer.
Roger, Pete,
okay.
that sounds good. Pete, are you on SPL right now? We can barely hear you. Can you check your B-Link?
I am on SPL. Is that better? I turned the mic up.
We can hear you. You just sound really quiet. You sounded perfect earlier. I'm not sure what changed.
Oh, that is. Oh, wait. How about that?
Oh,
yeah.
Hello.
Okay, I'm sorry. Let me back off. I think I figured something out.
Roger, you're loud and clear now. So you were just saying you wanted us to go around the room and give some introductions and backgrounds about who we have here.
If you don't mind.
Yeah, no, that sounds great. Science, do you guys want to start?
Okay, stand by. Let me tell everyone to tune in and listen up.
So to answer your question, yes, I guess we can start. Do you want to start, Chris, or you want me to start? Sure, I'll do it.
Go for
I'll
it,
bite the
senor.
bullet.
Yeah.
I'll wait till I hear from, make sure that we're okay. But as we're going, we see an umbellapathes, or whatever the new name is for that black coral.
Okay, folks. Everyone is tuned in and ready to hear about your amazing life stories.
Well, I hope they're not disappointed then. All right. This is, my name's Tom Leidig. I work for the National Marine Fisheries Service in Santa Cruz. I study deep sea fishes, corals, and sponges. Been doing this for about 30 years and been playing around with ROVs and AUVs and submersibles and all sorts of tech for most of that time. So it's just kind of a blast to be able to see all these things that you would never get to see otherwise. So is that a fun fact? Oh, fun fact is I am a certified sub pilot, so I can drive subs by myself. All right,
Whoa.
Chris, you can go.
Cool.
Hi, everybody. My name is Chris Caldwell, and I am the research coordinator. Like Tom, I work for the National Oceanic Atmospheric Administration, or NOAA. I work for NOAA's Channel Islands National Marine Sanctuary specifically, and we'll be surveying there in the next few days. And I'm the research coordinator, so I'm responsible for conducting and promoting and encouraging folks to do research and science that will support the management needs of the sanctuary. The sanctuary is a federal sanctuary, kind of like a national park, and it protects the waters out to 6 nautical miles around the Channel Islands off of Southern California, off of Santa Barbara area. The area we're in now, Santa Lucia Bank, is actually a proposed or nominated National Marine Sanctuary, the Chumash Heritage National Marine Sanctuary. And so we're exploring it to have a better understanding of the biodiversity that is common in this, commonly found in this region. Unlike Tom, I think I have the opposite background as far as this goes. He studied deep sea fishes and corals out here on the West Coast. I studied shallow water corals and fishes in tropics in Puerto Rico, Virgin Islands, and Florida and the flower gardens off the coast of Texas. And now in my new role, I'm having to transfer that knowledge into the deep sea, and am today acting as a student of Tom, who will hopefully get me certified in the deep sea world. So I think, yeah, I think that's a bit about me. I'll pass it over to the folks actually on the boat. Both Tom and I are doing this virtually. Usually we would be on the boat with the rest of the Nautilus team, but we're doing this all remotely from our living rooms. Handing it over.
Yeah, so I'm Ben Gracian. I'm a navigator and mapper on the Nautilus. So our role here is mostly, we have two vehicles in the water, Hercules, which is exploring and able to interact with the environment. We have another vehicle, Argus, that is directly kind of tethered to the ship. And so moving the ship around, we can move Argus, which enables Hercules to explore in a radius around that. And so we're kind of doing path planning and trying to optimize our coverage of the seafloor with the bottom time we have. And we also are making the maps from the shipboard, sonar and multibeam. So, and yeah, when I'm not here, I'm a graduate student looking at using kind of imaging and acoustic sensor platforms, kind of some new vehicles to kind of try to understand the distribution and habits of animals in kind of the vast three-dimensional space of the water column itself off the seafloor.
Yeah, I could continue on the introduction train. My name's Jess Sandoval. I'm part of the ROV team here in the Nautilus. Sit in Herc right now, so that means that I'm able to fly around Hercules, and yeah, fly is a term that we use both in air and underwater, I realize. But also we do a bunch of manipulations with the hydraulic arms that we have on the vehicles, or manipulators is the more formal way of calling them. And yeah, so we're responsible for not only kind of flying around, working with the navigators as well in order to get to where we need to go and take the samples that need to be taken. All right, Kim.
Really, Kim? That's it?
Kim has a whole bunch of experience in the ROV industry.
Yeah, haven't you been doing this for over 20 years? Do you want to say anything about your background?
We can't hear him on SPL.
Oh.
Oh, Kim, can you talk on SPL?
Hi, hello, I'm Kim Wallace. I'm part of the ROV team, currently sitting in the Argus seat. Came to this principally as an E-Tech. I do electrical and electronic work and for subsea instrumentation and ROVs. And I discovered ages ago, if you're in the business of fixing something and you know somebody's got an ROV, you've got it made in the shed. So, what else am I doing? When I'm not doing this, I'm doing this. I don't know, does that cover it?
That covers it pretty good.
Sure. This is Marley. I am one of the video engineers on board. Unlike